It's great to be here. It's been great just to kind of hang out for the last few days and kind of see what people are doing and how people are kind of working on targeted quantitation. And I think what I'll start off with is a little bit of what will be a little bit of a review, and I'll point out a little bit of our perspective in doing targeted proteomics, how we do targeted analyses, how we set up our assays. I think it's a little different than some of the things that have been discussed in the course so far. So I think it's good to have different kind of complementary viewpoints on how to go about doing this. I'll also talk a little bit about, as Eva was mentioning, about approaches that we've been trying to take to try to improve some of the selectivity of data-independent acquisition. I think it's an extremely exciting time in proteomics. I think we're really beginning to shift away from let's just see what we can find in our mixture and identify lots of things and generate a big list to actually really getting down and trying to nail down true, real quantitative numbers for proteins and peptides. And I think one of the things I've been seeing that people have kind of gotten out of doing some of these sky jams and these tutorials is that hopefully it's not as easy as everyone may think it is, right? It's actually kind of complicated to do these experiments. It takes a lot of practice. Okay, so this is when I started, my lab about almost 11 years ago. Now, this was kind of the gold standard. Everybody collected their data this way, right, by data-dependent acquisition. And it's one of those things I think that sometimes our field needs to go backwards a little bit before it can go forward, because this was a great tool. It's extremely powerful, but in order for us to move beyond the limiting factors of it, we need to kind of be able to think differently. One of the biggest challenges with this, and this slide doesn't do it justice, is these mixtures are extremely complicated. If you actually go and take a slice through the data here, like at any one mass of charge, you have hundreds, if not thousands of things that have the same unit mass. If you take a slice at any one point in time, you literally have hundreds of things at any one point in time. So if you try to do this experiment by data-dependent acquisition, you're always going to undersample to some degree. I kind of like this slide because if you actually start to do calculations on how many precursors there are in an isolation window, at any one point in time, there's an average. We tend to find about 121 peptide precursors at each point in time. And it can get up to about 300 at a point in time. And we have some LCMS gradients where we have 200,000 peptide-like features in a 1 or 2 hour gradient. So that's actually kind of a challenge in order to be able to find those. So there's no way you're going to collect 200,000 MSM spectra alone in an hour, let alone be able to actually get good enough data to be able to sample and identify these things. So, okay. You know this. You've been hearing about this all week. Targeted proteomics is one of the major solutions in order to be able to deal with this. So again, just to kind of review, here's data-dependent acquisition. You sort of select things based on the most intense features. You sort of get this semi-random sampling of peptides that throughout the mass spectrometry run. The one key thing to point out here is that you don't sample the same things over and over again. So you can't reconstruct chromatograms across your data. So you don't have the same precursor mass isolated at multiple points in times to be able to use anything, use the MSM data for really anything except for just qualitative confirmation that you're measuring what you think you're measuring. So, and also I'll also point out that this spends most of its time sampling the most intense features. Even though instruments are getting faster and faster and can sample lower abundance things, is it really a good idea to have an instrument method that actually spends most of its time sampling the most abundant things in the sample? We don't really need the most data collected on the most intense things. We actually need to spend our time, the most amount of time collecting the things that we're really interested in. Okay. All right. So in comes selected/parallel reaction monitoring. I think you probably have heard a fair amount about this during this week. And then basically the idea is now you have a precursor mass charge in time, and you sample reproducibly. And these are now greater selectivity than this, because instead of using like a 2 mass charge wide isolation window, we're using like a 0.7 mass charge isolation window. And we have these now sampled continuously throughout time, or at least time throughout when more peptides eluting. We have what we call an elution or duty cycle, which is the frequency at which we can sample that same precursor again in replicate points. And of course, we want to get at least 8 to 10 points across a chromatographic peak to define the peak. But this is an extremely powerful method, and now instead of focusing on the most intense signals, we're focusing on things based on a hypothesis or a set of targets that we're interested in. Okay, again, I don't think this needs, this is, we historically have done this on triple quads, although more and more frequently these types of experiments are being done on quadruple time-of-flight instruments or quadruple or Orbitrap instruments. And of course, the data now becomes chromatogram-centric. We're no longer looking at spectra as our main unit of measure. We're using chromatograms. And so we got it, and I've seen throughout this course, you guys are getting much better and much more used to actually looking at chromatographic data. Why do we really want to do this? Well, of course, the quantitative capabilities is the main reason. And if you actually begin to start thinking about this, here's a peptide spiked into human plasma. And if you keep blowing up this bottom end of the curve, you keep blowing it up, I mean, we have just unbelievable quantitative dynamic range and sensitivity. And I'm not saying that we have this level of sensitivity for every peptide. This peptide just happens to respond extremely well, but we actually can detect it down to low attomole range, and this is a previous generation instrument. We can now probably get about a factor of 10 better now with modern instruments, but we can detect them, and we get, of course, this great linear dynamic range. Now, there's not too many other analytical techniques that can get 4 to 5 orders magnitude linear dynamic range, but mass spectrometry on a triple quad can. All right, so what was the challenge in doing this? I mean, so when we began to start thinking about doing these experiments in 2005, it was really a tough thing to think about. I mean, imagine there was no software tools in order to build these methods. So what you basically end up doing is you take a protein sequence, put them into some sort of program that would calculate the potential peptides, you'd calculate the mass of those peptide sequences, you put those into an Excel sheet, and then for each of those, you take each of these individual peptides, put into another program, try to calculate the B and Y ion sequences, and then somehow you'd end up generating a bunch of transitions, and it was just a very complicated, cumbersome process. So in 2006, software for building these targeted methods didn't exist. Everyone was kind of carrying around these Excel sheets. They were hugely laborious. I remember I just wanted to measure one protein, and I remember spending 2 to 3 days just trying to figure out what the masses of the precursor and product ions were going to be in order to be able to do that experiment. So there was no software for the analysis of complex multiplex SRM methods. So this was the analysis of the data. And then there was also no software capable of cross-vendor data analysis. I think we take this for granted now. You can actually have some data collected on somebody's, Q-Trap 4000, another set of data set on someone's water Zevo instrument, and you can be looking at it all in the exact same time with the exact same peak integration algorithms, the same visualization tools. And we now kind of take this for granted, but this was something that blew people away when it was first happening. People could have one instrument platform, purchase another instrument platform, but they use the same tools for generating their methods and visualizing the results. And of course, this is also testament to the fact that a lot of your tutorials are using data from different instrument platforms. Okay, so we were lucky to get Brendan in the lab. Actually, people say, how did I find Brendan? I say to people, I didn't find him, he found me. He actually did show up in the lab asking if he could meet with me one day, which was really great. And so, and I kind of pitched this idea. There was actually a long discussion with Dan Liebler at Vanderbilt about what it would take to do something like this. And the key thing was, we need to find someone like Brendan, and we need to have enough resources to do it. So it was just barely enough to kind of get going, but it was enough to kind of prove that this was something that was important. So I still remember this. We started, Brendan and I started having a conversation. We started kind of listing some of the needs. It was needed to be protein and peptide centric. So most tools at the time were mass centric. You wanted to measure mass 752 and 326. What we really wanted to have was proteins and then peptides and then Y6. And so we wanted it something that a biologist or someone who was trying to make sense of this data could actually understand. Yes, you still wanted to be able to have the mass information there, but you wanted it to be peptide and protein centric. It had to have an intuitive document editor graphical interface. We wanted it to be a client tool. There were some web interface tools at the time, but one of the things that we wanted was the ability to be able to interface with the mass spectrometer. And so that meant it needed to be able to sit on a Windows operating system and to be able to read the data files directly. This was something that I didn't appreciate at the time. This is something that Brendan recognized instantly. I actually thought this was a lot of extra work. I'm like, even the vendors aren't even implementing this, but you wanted to do full undo redo support. And I think now people who see this in Skyline realize the value of this. It's really a document editor, right? And this is what Brendan said. He said, look, it's like an Excel document. It's like a Word document. You sit there, get to the point where you're making additions and subtractions to your transitions, and you want to be able to add transition, subtract it. What if you screw up? You deleted that transition. You really wanted to keep it. You wanted to be able to undo. So imagine writing your PhD thesis on a computer in a word processor that didn't have undo, right? It would be just painful. And so it's the same sort of thing here, right? And this was not a small feat in order to be able to add this capability. So another thing that we talked a lot about, and this became much more of a political thing that we had to overcome, was we wanted to be able to direct analysis of all triple quad vendor raw data without file conversion. When you guys install Skyline, there's a lot of vendor libraries that come with it that the vendors have been able to, that we've negotiated with the vendors to be able to distribute those libraries with Skyline. And that was, as Brendan says, it was, I think he said it was 2 years of his life trying to figure out how to do that and get all the bureaucracy handled in order to get the licenses appropriate for this. We wanted to be able the ability to use data from shotgun discovery data necessary. It needed to be free and open source, and it needed to be developed professionally. One of the things that Brendan was saying at the very beginning was it needed to have a support board, and when people had questions to it, we wanted responses within 24 hours. If not saying that we'd have a fix for it, but even just acknowledging the problem and how it was going to be tracked and kept track of. So this was something that was again, I think very different from what a lot of other academic labs have done. And while I'll tell you right now, while I've been doing, I would call bioinformatics for most of my scientific career, I've never done any software engineering, and my labs I had no experience with doing any software engineering. So it's great that Brendan kind of came in who had a lot of the software engineering experience could come on and take on this project. So I think we're all really excited about the growth. We're I think over 30,000 installations by now, but we were excited when we broke 100 in a given month, and it was. But of course now we're doing it continues to grow, and Brendan was telling me just the other day that we just hit 11,000 startups of Skyline in a single week. So there's definitely a lot of people using it. Okay. So I figured I'd start simply and take you through a project that we have going on in the lab. This is a work in progress. This isn't something that we have completely solved. And so we're trying to get the manuscript together right now. This is an SRM assay that we're doing. We're trying to come up with a targeted, what we're calling a wellness assay. This is an assay that, in theory, would be done in the clinic, in the hospital. You'd go, before you go to your doctor's appointment, you'd be sent an envelope in the card with a blood card. You'd prick your thumb, stick it on the card. You'd send it out to a clinical lab, and then when your doctor goes to see you, he would have a list of all the abundances of about 50 proteins in your sample, and it would give a sense of, and it would be something that could be done inexpensively enough that you could almost do it once a month, and it would be something that your physician could track your health over time. And it wouldn't necessarily tell you exactly what's wrong with you, but it would tell you whether or not you're sick or getting sick, or allow you to see what your baseline levels are for certain types of proteins. Okay, so, and we want to do it from dried blood spots. And one of the main reasons here is that it's easy to ship around. They can be stored at room temperature. And there's a lot of things actually in red blood cells that are not in plasma. And it's just, again, very easy. You don't have to worry about having a phlebotomist draw blood or anything. It can all be done just by at home. So the questions we're developing a target assay to measure gene products via peptides. First are our challenges. One, what peptides generate strong signals, chromatograph well, are unique to, or stable and robust over time. So we kind of have a little bit, I can think of a different idea of what an assay is than some other people do. What transitions provide a sensitive and unique measure of that peptide. And also we're trying to use this measure of this peptide abundance. It's in dried blood spots as a proxy of the protein that is historically done by an ELISA. So we actually will do a step where we validate that each peptide measurement does or does not correlate with the standard clinical assay that's being done in the clinical lab. So if we decide, okay, well, what proteins are we going to pick? So if you actually go through, there's this fantastic textbook that provides about 100 protein assays currently done in plasma in the clinical labs. Now these are all done using completely separate different types of assays, and we wanted to see how many of them could we get done in a single assay without any enrichment in dried blood spots. And so all the ones listed here are the ones that are currently done in the clinical lab. All the ones in red, we had cDNA clones that we could make recombinantly very easily and inexpensively. I'll talk a little bit about that in a second. So we made recombinantly all the proteins in red, and those were our starting points for choosing 50 proteins that we're going to then use to make a single clinical assay of. And we want to have this down the road in less than 60-minute gradient time on a triple quad. So one of the things that people say is, well, we'll just use discovery data in order to find which peptides we need to measure. And one thing that we found a while ago is that this isn't necessarily the best thing to do. And so here is a plot of, in red, is in ApoB, this is an enormous protein. It's about 500 kD. It's the main protein in bad cholesterol and LDL. And people have shown it's a much better predictor of cardiovascular disease risk than LDL itself. So, of course, there's one of our proteins in our assay. SRM peak carry for every single peptide that's measured is shown in red. And then the ones that we detect in plasma from just doing DDA experiments from 12 separate independent DDA runs are shown in purple here. But if we were to choose the peptides based on the ones that get sampled most frequently, this would be the one out here, which is more than 100-fold less intense than the actual best responding peptides that are done by SRM. In fact, I think the top 10 peptides responding by SRM are not even found in DDA runs of plasma. At least not routinely, unless you do a lot of fractionation. And then, of course, the ones that are here, you'd be picking ones that, again, they're 10-fold to 100-fold less intense than other ones. So this is actually pretty interesting, and we find this over and over and over again. There's very little correlation with spectrum counts with the actual relative abundance of the SRM data for an individual gene product. So we've actually gone through and actually tried to do choosing our peptides based on the SRM data itself. And there's actually a process within Skyline that was kind of built to kind of optimize this, which we call this kind of refinement process, where you could actually measure a very large number of peptides by SRM in a sample matrix and then refine it to the top responding peptides. So this is just an interesting observation. We've made this plot for 760 proteins at different times, but it's definitely, it's a very common trend. So here is the way that we do it, our general refinement workflow. So we start here by recombinant protein production, and I'll talk a little bit about that. So this all got started actually by a seminar from Josh Labar. Josh Labar from Arizona State University. He makes protein microarrays, and on this microarray he expresses 10,000 proteins on a chip. And I saw this talk, I'm like, this is absolutely incredible. If you can produce 10,000 individual proteins, he's proving that they're individual proteins on a chip. Why aren't we making those 10,000 proteins individually ourselves and then using that information to help us develop quantitative assays? All of his proteins are GST fusions. And so we had this really ambitious graduate student in the lab, and he wanted to study transcription factors. So he started off going through and making all transcription factors. We use this to make these plasma assays. So basically, we take his cDNAs from Josh Labar's lab at this DNA SU. We expand, purify, and sequence the cDNA clone. And we use an in vitro transcription translation system. Pierce came out with right at this exact same time, came out with an incredible kit that solved a lot of our problems. It uses human expression system. We enrich by glutathione sucrose, since all of them are GST fusions. We now have our gene product of interest fused to GST, and GST itself is actually 30 kD. It's actually a big protein. And while we don't have this protein pure, we can actually determine exactly how much is there because we actually have made peptide standards to this GST, and we've done calibration curves. In essence, we've had a 100% success rate in making proteins in Josh Labar's cDNA clone library, and we make between 25 and 500 femtomoles per 25 microliter system of IVT, and it's less than $20 a protein to make. So for less money than it takes to make a crude synthetic peptide, you can make the entire protein. Now it does require a little bit of doing a mini prep and express your protein, so there is labor involved in this. But I'll also point out too, we had a single graduate student make 760 of these proteins in 2 weeks. So it's definitely feasible to scale this. We've now made every transcription factor, every kinase, and we've made almost every protein that's ever been used in a clinical plasma assay before. So and of course, the goal of this is to build chromatogram libraries and have it all available on Panorama that people can use this information. Okay, so the next thing is we monitor all tryptic peptides by Skyline. This is part of the refinement. We have some basic rules in which we kind of limit things. We just basically stick to doubly charged peptides in general. We don't fuss that much about triply charged. And so for a protein like ApoB, this is kind of what the run looks like. There's a lot of peptides in here. We're trying to see what peptides give the best response or not. And here again, apoprotein B100, you kind of rank them by the most intense to least intense. It's actually done using SRM data. It's done on the actual protein of interest, etc. So then the selection of transitions and calibrating retention time is the next step. We basically use this since we've measured every Y ion from Y3 to YN-1, and there's a big reason why we stick with Y ions right now for tryptic peptides. We wouldn't do it for other peptides. And I think if you were using a SciEx triple quad, you'd probably want to include some B ions. But in general, with the Thermo Fisher triple quads, B ions, except for the B2, they're almost non-existent. Or at least they're much lower, reduced in intensity relative to the Y ion series. So basically, you have one peptide, and you basically get this sort of fingerprint for every single peptide that you're measuring. And as Brendan was bringing up earlier, instead of using now the spectrum libraries, Brendan came up with this concept of chromatogram libraries, because what we're really doing is generating a library based on our chromatographic data. And again, we use, for IRT calibration, we use a protein standard called ApoA1. It's the main protein in bad cholesterol, and we use a 15N analog of it. And it's commercially available. It's really inexpensive. This protein's a rock. And it's it gets digested with every single sample. It's in the mixture. We use it for QC. We use it for IRT calibration, and we also use it for as an internal standard. And I'll explain how we do that too. Okay, so we use this to calibrate the retention time. So this next step is very important, and I don't see this in too many other people's presentations and how they choose their peptides. But we've found this to be incredibly important. So peptide stability analysis. So we make proteins in 96 well plates, and what we do is it takes us about 72 hours to go through all injections of those 96 individual proteins, and then we go and inject them a second time, and we compare the intensity from the second injection from the first injection. They're all stored at 4 degrees in an autosampler. If the peptides are still good at the second injection, then we say that they're probably pretty good stable peptides. If they've dropped in intensity a lot, then we begin to wonder whether or not we should use them. So I'll give you an example here. Here's the first injected in immediately after its production. Here's 72 hours sitting in the autosampler at 4 degrees. You say, okay, it's dropped a little bit, but it's not dropped that much. This one, it's dropped a little bit more. This can be a little bit borderline, but this one, we would not want to use this peptide, right? And there's lots of reasons why this happens. Sometimes it's deamidation, sometimes it's oxidation. We actually have had a hard time tracking down why some peptides seem to disappear in intensity, and other ones, we think some may stick to the tubes. It's just, it's not clear, but we don't want to be building assays off of peptides that disappear in the tube over a long period of time. So, and it's not like they even disappear very selectively. We just don't understand it, and it's clear that some peptides don't stay around or aren't very stable. So we do this as a main step in all of our assay validation. So we don't necessarily choose the most intense peptides, we choose the most intense peptides that are stable as well. Okay, so validations of transitions to the matrix. Just because you have good peptides in a standard doesn't mean that those transitions are going to be good in the matrix, because if you don't just want intense peptides, you want intense peptides that are absence of interference. So here it is from our standard. We have our IRT, so we can help pick the correct peptides. We have our dot product information, and we confirm that those same transitions, in this case now it's a dried blood spot sample, is actually good. And we do the same thing again for every single peptide that we've gone through, and it's actually been pretty exciting. So here's the last other step that we do is finally, if we get down to 5 peptides that we really think we want to do, we'll do a digestion time course. So here, you look here, this peptide, we do a digestion over time. And you can pretty much say, look, if we choose a time out beyond this 720 minutes, we're pretty comfortable that the intensity is going to remain pretty constant throughout the entire digestion. This is a good peptide, and we're going to stick with, we'd probably use this one. There's some peptides that are like this. So this, again, would not be a good peptide to use because depending on whether or not you had your digestion here or here or here, you could actually get very different signal intensities. And this one, I'll point out, is actually a protein from our internal standard, ApoA1. So we actually do have a 15 analog of it, and the 15N analog looks identical to it. So if you actually had the stable isotope labeled protein for all these, it would be fine because it would actually account for this weirdness in the digestion. But if you don't, for that individual, for a protein standard for every case, it's actually not going to be a very good peptide. So again, it's just good things to worry about. I also told you what we're trying to do is to come up with an assay that we believe is a good proxy for that what's used in the clinical assay in an actual ELISA assay. So here what we did is we had 42 matched samples from serum and EDTA whole blood dried blood spots and also just capillary dried blood spots. And we have on y-axis we have our area under the curve ratio relative to what we call a global standard. I'll talk about that in a second. Relative to the clinical labs immunoassay. This is done by Andy Hoofnagels. And in this case, there's only a subset number of the proteins, but this peptide correlates fairly well, and this peptide correlates fairly well. There are some peptides that don't correlate very well with the established clinical assay. Now, the problem here is which one's right. We, of course, believe our data is right, but it's still going to be very hard to actually take that forward and have physicians use that as a proxy for the clinical assay when it's disagreeing with what the current standard of care is. So we're currently using this as a way that we want to demonstrate that our results are well correlated with the existing standard of care for those patients. Again, we do that for, and again, some peptides for the same protein give you different results. So it's important to do that for each peptide. Okay, normalization and calibration. So we have 50 proteins we want to do. We've got 4 peptides per protein. That's 200 peptides. We don't want to order 200 stable isotope labeled peptides that have to be spiked into every single sample. So can we get by with just spiking in one protein standard that we use for IRT, QC, and also just as an internal standard? And so again, this is where we use this apolipoprotein A1. We had done a fair amount already using this as an IRT standard, and we kind of did a fair amount to kind of compare that the peptides had a nice retention time distribution relative to the Biognosys IRT kit. And it actually gives a slightly broader retention time range. It's economical to use, and it's a pretty really robust protein. It also gives us good QC that the digestion worked okay, because if you don't see these peptides of a certain intensity, then it's kind of a confirmation that the digestion didn't work ideally. So in the clinical environment, this is pretty important. So this takes a little bit of showing. So here we have 2 peptides. So if you look in the columns, this peptide and this peptide are the same. What's different between the A and C and B and D is that A and B are done with a stable isotope labeled peptide of that individual peptide sequence. When in C, we're actually measuring this peptide relative to a different peptide. So it's a sum of, I think, 3 different peptides from our apolipoprotein B standard. And so it's not actually a direct standard of this. But if you'll notice, we do have some loss in linearity, and we do have some slight loss in precision, but it's very, very small. And so, and we weren't the first people to show this. This has been published already in clinical chemistry. It's showing that basically you get the exact same results if you take apolipoprotein B and measure it relative to its own 15N apolipoprotein B standard, or relative to a different protein standard. And so Andy Hoofnagel has this great paper in clinical chemistry confirming that, and that you don't actually need to have a stable isotope labeled peptide for every single individual peptide. It's nice when you do because of all the qualitative stuff that you've learned throughout here, but as far as quantitation goes, you can get pretty darn good quantitation by using this 15N apolipoprotein B apolipoprotein A1 standard. I know I'm talking quickly, but I figured that there's a lot to kind of cover. So this is, anyway, was pretty exciting to us. And we have a product. We've also done this in saliva for about, I think it's about 30 proteins, and basically shown that the precision that we're getting for using this global apolipoprotein A1 standard is almost as good. It can't be as good, but it's almost as good as using a stabilized labeled protein standard, and it's definitely as good as using a stabilized labeled peptide standard. Okay, all right. So that's basically how we go about building targeted assays in our lab. It takes a lot of work. The validation process is huge. Just because you have some peptides from a discovery experiment doesn't necessarily mean that you can translate and build that into a targeted assay. One of the things that's been kind of exciting to see, and from something that Brendan I don't think we'll get to today, is we have this web server called Panorama. But one of the things that's being used for is the National Cancer Institute in the United States. They have the CPTAC program, and they're building it to store an assay portal. And they actually go the extra step beyond what we're doing in a lot of their validation in actually confirming the actual level of quantitation and the actual linearity for every single peptide. But I still think what we've done is close to that. It's just not quite yet to the same rigor as the CPTAC assay standard. Okay, so if anyone studied these assays knows measuring tens of peptides by SRM is okay, but once you start trying to measure hundreds of peptides, it gets to be challenging. You're going to have to talk about scheduling. These are complicated experiments. You're talking about picking transitions. When if you have an instrument that could measure all the transitions, you don't have to worry about picking transitions at the initial development stage. You can just measure them after the fact. So we began to start thinking, as a lot of other people did, is that, okay, at some point in time we're going to need to multiplex the MSMS in order to be able to measure, to be able to get many peptides in a single gradient without scheduling. So we had begun to start thinking about using wide windows to capture multiple things simultaneously, and use ion trapping instruments where we measured all the product ions in a single scan. We started doing these things back in 2004 or 2005. The instrument hardware just wasn't up to standard, and so we kind of put it on the back burner for a while. But after then, we started looking at the data from a different perspective with Skyline. It began to make a lot more sense. We started to revisit again using the latest Orbitrap mass spectrometers. So if you want the selectivity of targeted SRM with the breadth of data-dependent acquisition, so if we want to have a new peptide to target, but we don't have to go back and rerun the experiment, we basically, what other people have called, want a digital molecular archive of all samples. And there's two things that I think that kind of come out of this. So one is, we've seen in our lab and other labs, is you build an assay for 100 peptides, you measure 100 samples, and then once you're done with that, somebody else comes back to you and says, oh, can you measure this 101 peptide? You're like, ugh, we don't even have those samples anymore. And so yes, those samples would be perfect to measure that extra peptide, but yes, going back and rerunning them all again is just impractical. So you wanted to be able to have a way where you could actually go collect all the data once and be able to continuously mine it over time. The second thing is, which I think is something that's really critical to this, is that in theory, it should be a great digital molecular archive of all the samples. But in order for this to happen, we need to prove it. So there are now becoming published data sets out here, and this will not become a useful method in this regard until a different lab goes and analyzes data from an already published experiment where that lab did not analyze the target protein you were interested in, but they used your data to generate a new finding that was missed in that data set. It's something that, in order for this to truly become true, somebody's going to have to do this. I really encourage people to go about doing this. There's now a fair number of data-independent acquisition data sets out there, SWAT data sets out there. You should go and try to reanalyze it, publish a paper on finding a new protein or new peptide that's changing in that data set that was not done in the original data set. It's not going to be until that happens over and over and over and over again that this becomes a very useful and valuable method for the community. Okay, so I talked about beforehand, these were kind of two general approaches. Data-independent acquisition is kind of our way of thinking about doing this. And here, instead of having these narrow windows where we have hypothesized targets, like has already been mentioned yesterday, we're choosing wide windows that cover these big blocks of the mass range. We've got a couple of different ways of illustrating this. So I was in the lab when John Venable was first trying to do this. He started trying to do this around about 2001. Failed miserably on an LCQ, and then finally got some decent data in 2004 when the LTQ came out. But the experiment design was fairly simple. It was using, I think, 10 to 15 master charge isolation windows. The instrument scanned at about 5 hertz at the time, and so you could cover basically 100 master charge range in a couple seconds. And so if you have your chromatographic peak with this target peptide here, you can extract chromatograms, and you have multiple different points across the peak that you can regenerate the chromatogram. Another way to show this data is that you have this precursor range, and in here you can look at it in a heat map type view, and you can see that you get these product ions that kind of co-vary over time, and these are all the same product ions that belong to the same individual peptide precursor. This was all done on a low-resolution linear eye trap back around about 2004, 2005, and you see this blank region in the spectrum because we were using resonance excitation CID, so that we don't see that anymore with HCD or beam CID. And so you can actually then use this data to go and pull out either an MS spectrum by taking a slice through the data this way, which is what most people were trying to analyze at that point in time in 2004, or you can slice through the data this way, which is basically what Skyline does and generates a precursor product ion chromatogram, XIC, and this is basically the equivalent of like what you get with SRM type data, except it's a lot less selective because you have a wide precursor window, and in this case it's low resolution product ion data, so we don't have the greatest product ions resolving parts either. So this is another way of looking at it. There's this isolation window, and you can go and generate, you have the entire spectrum at every point in time, but you can extract the individual product ions that belong to your target peptide of interest. So in Skyline, we represent the data like this, and of course, we don't just have the product ions, we also have the precursor ion data. So we can make, and this was just being shown just a few minutes ago, where you can use the combination of these to confirm the charge state, the monoisotopic mass, and also the product ions. Okay, so this shouldn't be a surprise, but wide isolation windows results in high interference. So we've at times struggled a little bit sometimes with 5 mass charge wide windows. We've actually struggled with 2 mass charge wide isolation windows at times. If you look at DDA data, 25 to 50% of all DDA data with 2 mass charge wide windows has 2 or more components in the isolation window. So 5 mass charge wide data has substantially more than that. 20 mass charge wide data has even more than that. So when you start looking at 5 mass charge windows, here's a product ion spectrum. It's definitely a mixture of multiple things. We go to 10 master charge. There's actually components in the isolation window that are 100 times more abundant than the target that we're actually interested in. So it becomes actually hard to look at the spectrum, but looking at the chromatograms with enough resolving power, you can actually deal with it. But you can definitely see that there's an issue in detecting this peak relative to a smaller, narrower window. Just to kind of give you, we did some kind of theory based on, this is just Saccharomyces cerevisiae. What we did is we took every single peptide. We have a relational database in our lab where we took every single peptide we've ever identified by Saccharomyces cerevisiae. We looked at their relative retention times. We looked at their isolation windows. And basically, if you have a 2 mass charge wide isolation window, majority of them by DIA would have none. We have about 20% would have 1, and some would have 2. And about 20% of the ones that have 1 would have 2. If we look at the 4 mass charge isolation window, again, you have 50% have none, but 35% now have 1, etc. When you talk about 10, now we're looking at majority of them have 1 or more. When you start looking at 25 or more, you start to have almost all of them have, some of them have 10 or 15 components in the isolation window. So all this comes down to the fact that picking peaks now becomes hard. The quantitation is actually still pretty good regardless because you can pick transitions without interference. And I hope that some of the tutorials end up showing this a bit, but it comes down that picking peaks becomes a challenge. And I think Brendan might have shown a slide very similar to this. You can look at these chromatograms, and this is the precursor ion chromatogram, and here's the product ion chromatograms. And you're trying to find what point in time is the peptide you're interested in. It's actually this point in time. And the reason why we know is the exact same thing that Brendan was bringing up yesterday, is we ran interspaced with the DIA data a DDA run where we had 2 mass charge isolation windows. And when you look at these cyan lines that are showing up here, they're all kind of very close to this peak. And we look here is the precursor ion chromatograms, and here's the product ion chromatograms. But you see in the precursor ion chromatograms, we don't really have enough selectivity to do quantitation, but we can do it for the product ion data fairly well. So it's actually pretty good. When you open the windows even more, we get, so in this case, it's 10 mass charge isolation window. This peptide is fine. We just tailing, of course, doesn't chromatograph that nicely. This one, a couple of the transitions have interference. But again, it's not really that big of a deal for quantitation because you can just drop those 2 transitions that are causing you problems. And we've been thinking a lot about having automated ways to drop the transitions, et cetera. And it's going to take a little bit of time to implement them. Okay, so one of the things that we've been discussing a lot within our lab is this idea that that data-independent acquisition is all about balance, and sometimes people in the lab say it's about sacrifice. It depends whether you're a glass is half full type person or glass half empty type person. We've kind of chosen in our lab to do data-independent acquisition between 500 and 900. And people ask us why. I mean, okay, ABSciX and other labs have gone through and used this very wide range of at least 400 to 1400, if not larger. And our goal has mostly been, we just need to get a couple peptides per gene product. And so, it is the most dense region in the mass range where most of the doubly charged peptides appear. It's where the best responding peptides tend to appear at. Yes, there are cases, especially if you want to get phosphopeptides, that you may need to get outside this mass window. But it's like the greatest good for the greatest number, right? So do you want to sacrifice some of your, do you want to add a greater range and widen your windows and decrease your specificity a bit to get to windows to a larger mass range or lower mass range for a few peptides, or do you want to make it so that the majority of your peptides in this 500, 900 mass range end up responding the best? So this has been our choice. Some people, I think, go from 400 to 800, but I think most people are settling now in this 500, 900 range. So if you have 20 by 20 mass charge wide isolation windows, you're looking at basically an instrument with 10 hertz, about every 2 seconds sampling this. So, if you have a 30-second wide chromatographic peak, you've got plenty of points across the peak. We can improve the selectivity by doing 40 10 mass charge wide windows. That, of course, would mean we sample it half as often. And you can do 5 mass charge wide windows, but that would get to the point where you probably wouldn't want to use the product lines for doing much quantitative data, because you're not going to be able to define the shape of this chromatographic peak very well based on the number of sampling points that you have. So one thing I also want to make sure that you don't rule out is the ability to do multiple injections. This is still a fairly common workflow in our lab. We will make at times multiple injections to cover the full mass range. It's less throughput, but you get better data over the range that you're interested in. So it's something that shouldn't be ruled out. It's a very powerful way of kind of handling these things. We've actually even done some cases where we do 17 injections with 2 mass charge wide isolation windows on older generation instruments, just because you get really good data and you cover the entire mass range, and you don't have to worry about, you can sample the points multiple times across the peak. Okay, so we've been working in this area for a while, and we've been trying to think of ways to improve this for a long time. We want to be able to sample things fairly high frequency. We want to be able to cover a breadth of a mass range, at least this 400 mass charge range. So we want to be able to get to like 20 mass to charge in a single mass spectrum. But we wanted to have the selectivity of about 4 mass to charge. And people ask, well, why 4 mass to charge? There's this critical mass unit in biology. It's CH2. And it's the main mass unit that we have to have a mass range that's better than that. So that's 14 Daltons. So doubly charged, it's 7 mass to charge. Triply charged, it's 4 and a bit, a little less than 5 mass to charge. So we'd like to be able to have an isolation window that's better than CH2, because it's also, if you're looking at a SNP, most single nucleotide polymorphisms cause a change in CH2. Like valine to leucine, serine to threonine, aspartic acid to glutamic acid, they're changing CH2. And if you get better than the CH2, you'll also cover things like oxidation fine. And the only thing we won't be able to cover very well is deamidation. So, but pretty much every other modification we can handle. If we have a 4 mass charge wide isolation window, it's actually the ideal thing if you can think about it. We want an instrument capable of doing 4 mass charge across the entire mass range. Of course, that's not really possible. So this is the way we decided to try to do this. I remember seeing a talk from Makarov about 2 years before the Q-Exactive came out, kind of showing a spec on what this instrument was going to look like. And I knew right then that this was going to be an interesting hardware platform to do these experiments. Because in theory, you could use a quad, isolate a mass, fragment it, store the fragments, isolate another mass, fragment it, store those fragments, and then measure one mixed spectrum in a single spectrum. And I'll show you in a little bit why we wanted to do this. So on the Orbitrap, on a Q-Exactive, this is the way basically the time works, right? So you have time, here's the scan, and then for the next scan is you spend a certain portion of the time filling the Orbitrap for the next scan, for the C-trap for the next scan. And what we basically wanted to be able to do was multiplex this with 5 separate windows. We would lose no time, but yet in theory, if we could demultiplex this back again to the 4 separate individual mass ranges, we'd be able to solve our 4 mass charge wide problem with a 20 mass charge breadth and mass range. So here's basically the idea. This was the vision, at least. 100 4 mass charge wide windows. You can't really do that in a chromatographic time scale, unless, of course, you had an instrument that was 50 hertz, which we don't currently have, but they'll be coming. So the idea that we had was to fill with 5 separate windows, collect in 1 spectrum, then randomly select 5 for the remaining 95, collect 1 spectrum. I know that this is actually complicated and makes your head hurt in thinking about it a little bit. And then so you have 5 more in the remaining 90, and you keep doing this until there's only 5 windows left. You fill it again and collect on the 20th scan. And then in the 21st scan, we refill the list again, and we do the experiment over. Now there's a couple things that's kind of critical here. In scan 20 and 21, there's only 1 mass range that's shared. So if you have the same fragments in 20 and 21, we kind of make the assumption that they must be coming from the same isolation window. If they're different, then we must assume that they must be coming from the other 4 isolation windows. And we can use some old analytical chemistry techniques that are very similar to like Hadamard transforms to be able to demultiplex this. And we had tried Hadamard transforms back in like 2004, 2005 using overlapping windows. And Jared Egerton had this great idea to use this non-negative least squares, which was just very, very simple solution. And basically his idea was our problem with the Hadamard transforms was that some of the peaks were coming negative. And he was just like, I'm just going to force my linear algebra that everything has to have a positive correlation. We can't have a negative signal. And it made everything work so much better. Okay, so our cycle time is in the order of 2 to 3 seconds. And it works pretty well even with the first generation Q-Exactive. But now basically this is the way the data looks. We have data that's for each mass. In theory, we could have a combination of 5 different isolation windows. We need to be able to demultiplex this to the 5 individual components. And this is basically the way it works. I'll spare you a lot of the math that goes into this. But basically, here's the data that looks undemultiplexed. There's basically 20 mass of charge worth of isolation windows in each scan. We can demultiplex it to look like this. And if you look here, this is a much easier thing to pick the peak over here than it is over here. Here's the data again blown up. And again, quantitatively wise, we could just remove the transitions that show interference. But again, it just looks a little bit nicer and it's easier to deal with when you do the demultiplexing. So if nothing else, it definitely helps as far as the qualitative peak picking aspects of these experiments. We'll just have a few more examples. So here's 10 mass charge DIA. You're like, okay, well, which peak do I pick? MSX, which covers 20 mass charge in each spectrum, demultiplexed. Of course, it's much easier there. And here's just a blow up. Again, you could use this data fine for quantitation, but it's much easier for picking the peak to use this. Here's just another example, 10 mass charge DIA. Here's 20 mass charge in each spectrum demultiplexed, and again blown up here. Again, you can use this fine for quantitation if you know which peak it is. And here's, but it just looks, it's just easier to deal with. People, we do collect the MS1 data at the same time, and one of the things that we had shown, and we continue to show, is that if you have a very complicated matrix where chemical interference becomes your limiting factor, we do have a lower limit of quantitation and a lower limit of detection by MSX. However, in a simple mixture, we don't. And I'll talk about that in a little second. In a second. Okay, so I want to talk a little bit, another example I think that was brought up, yesterday, which is an issue of things like PTM modified peptides. Here's a peptide, and it's oxidized form of that same peptide. Almost all of the product ions are shared. In here, they both fall in a 10 mass charge isolation window. And if you do a 20 mass charge with the data and demultiplex it, it demultiplexes this peak away. I'll point out too, this is an earlier form of our demultiplexing algorithm where the peak got distorted a lot more. We actually started using Svirsky-Goulet type weights, and it improved it dramatically. But again, you still qualitatively see, in this case, we can eliminate this peak from being a candidate because it's not with the precursor is outside the 4 mass charge isolation window. Even though this is a 10 mass charge worth of data, this is 20 mass charge worth of data in each scan, but we demultiplex it to 5 4 mass charge wide data. Okay, so people ask me all the time, well, do we use this MSX approach in our lab every single day? No, we don't. And they say, well, why not? And if why not, then why haven't you solved this problem and what is the problems that you're running into? So the big problem is not the specificity. The specificity is great. The problem is that we just don't have enough ions from time to time. So if you actually look at retention time windows in an Orbitrap, we basically give it a target where we want to put 500,000 to 1 million ions in the Orbitrap, and we give it a maximum amount of time that will allow that to happen, and then we figure out how much the time does it actually hit the maximum inject time versus how much time does it actually trigger at a value at a less time. So here's across the retention time, and here is majority of the time we're pegging at the maximum inject time, so we're just never hitting that target. And there's a little bit of time where the most intense peaks are coming out where we do end up, filling the Orbitrap for some of the scans. You can also plot this another way. We were shooting for 500,000 to 1 million ions in the trap, and we're underfilling dramatically. So we're not hitting that many ions. And since we're doing 5 4 mass charge wide isolation windows, we're definitely underfilling. Now, there's another complicating factor in here. I'll bring up a second. So the Orbitraps are even getting faster. The Fusion and the Q-Exactive HF we use in this high field Orbitrap scan at an amazingly fast rate. So if you look here and he said, okay, we're going to just let the Q-Exactive run at the scan rate at 15,000 resolving power, it can easily scan at 25 hertz, no problem. And actually, if you put it at 7,500 resolving power, it would actually could scan it at 50 hertz without too much problems at all, and you can still get 2 ppm, 5 ppm mass accuracy with external calibration over months. And but the downside is that we just wouldn't have any ions to deal with in an experiment. So if you sit there and do something a little bit more practical and say, look, we're going to set this to being running at 20 hertz, which is basically 40 10 mass charge wide isolation windows, it's going to end up being about 17 milliseconds is the maximum time we can allow the C-trap to fill. 17 milliseconds isn't much time to do much with. And so even with, I think, the Q-tops, is people integrate the peak for a fairly large amount of time, at least 50 milliseconds. So in this case, 17 milliseconds isn't much time to accumulate a signal or accumulate ions to measure the signal. And so what we often do is drop our increase our resolving power even further to 30,000 resolving power. We go to more about 10 hertz where we can fill up 60 milliseconds. So it's kind of interesting. People talk about this new Q-Exactive HF, which is an extremely powerful instrument that can run at 20 hertz, no problems. But if you run it at 20 hertz, you'll have almost no sensitivity. And it's better to actually run it a lot slower at higher resolving power, where you can actually then fill the load trap. And there's the other complicating factor, is that if you say, look, I'm willing to take 100 milliseconds maximum inject time, and if you're multiplexing by 5, you can't spend more than 20 milliseconds on any one of the 5 windows. So our maximum inject time was actually 20 milliseconds for those MSX experiments, even though our Orbitrap transient was collected for 100 milliseconds. Okay, because you're filling these all in series. Okay. So this brought us back actually to an experiment we did in 2004. Mike Hoopman had done it originally. We just hadn't found a good way to demultiplex the data. And in collaboration with Dario, Amadai, Parag, Malik, and Olga, we met, we thought it may be time to take the stuff that Jarrett had learned to improve it and kind of work with them to kind of improve this experiment. But we had collected this data on this in 2004, 2005. And so if you have 20 mass charge wide data and you have 2 components in the isolation window, showing here, this is our target where the checkbox is. This is the one that we don't want. This is interference. And we're showing here the blue and red scans. We could either collect data at half the window size and collect half the breadth of window, so only 200 master charge, and discriminate between these two. The other option is to offset the isolation windows in alternating scans. So in the first scan, you measure it in the 880 to 900. In the second scan, it's measured from 890 to 900 to 910. And so the peak that belongs to our target of interest is here. And now the peak that's not in our target, the other half of the window becomes like the sawtooth pattern. It's kind of going in and out of the isolation window. And Dario saw that what we were doing here, and that the stuff that Jarrett had done with the demultiplexing beforehand, and saying, I can demultiplex this using the same general approach that Jarrett had done previously, with some improvements. So basically here is 20 master charge wide DIA. Notice there's lots of peaks with the same product times. Here's 20 master charge with overlap. Notice here there's a sawtooth peak. Here's another sawtooth peak. There's other ones that are not. They're kind of quite smooth. These are the ones that are in the correct half of the 10 master charge wide side of the window. These are the ones in the other half of the window. And when you apply Dario's demultiplexing, that's what the data looks like. So again, I'll go back and forth. This is 20 mass charge DIA overlap demultiplexing. Here's it without the demultiplexing. And then if you go and collect the data at 10 mass charge, which is basically the best case scenario you can do, that's what the data looks like. So basically there's almost no difference in data from doing 10 mass charge wide data independent acquisition and 20 mass charge data independent acquisition with overlap and demultiplexing. And I'll point out that Skyline just handles these data. It doesn't matter if you do it on a Q-top. It doesn't matter whether or not you do it on an Orbitrap. If you collect data like this and you use the isolation schemes that are within Skyline, it will demultiplex this data. And we would actually love people to collect data on Q-tops and tell us it's actually working correctly for the Q-top. Because in theory, it should work. There's no reason why it should be any different. But we haven't found anybody who's started collecting data yet this way. And in our lab, we've pretty much decided that we're not going to collect wide windows without offsetting it anymore, because we can always gain back that specificity, and we lose essentially nothing in our quantitative precision and performance than not overlapping the data. So one of the things that kind of helps with the overlapping, although we've done it without it, is the fact that we have a fairly uniform isolation window on the new Q-Exactive Plus or the Q-Exactive HF. And so it's so signals on the left-hand side and the right-hand side have a very similar transmission efficiency, but we're able to do this beforehand regardless. So even with the old Q-Exactive, it still works. In fact, all of our data for publication we're still trying to get out was all collected on an old Q-Exactive, and the data I just showed you was collected on an old Q-Exactive. So people often ask us, what do we use as general starting experiments? Is there a one-size-fits-all experiment? I don't think there is. I think depending, every experiment is going to depend on what you need, and if you have a simple mixture of protein complexes, wide windows are fine. If you have a really complicated matrix and selectivity is a problem, then you're going to want to use maybe even something like MSX. So there's two places we tell people to start at. If your issue is sensitivity, we tell people 20 by 20 mass charge isolation windows. You get 10 mass to charge isolation after demultiplexing. Your fragment ion selectivity is 30,000 resolving power, and you can have a 60 millisecond maximum inject time. And then if your issue is selectivity, we tell people now to do 4 or 5 mass to charge MSX data. You have 5 mass charge isolation after demultiplexing. You get 30,000 resolving power in the product ions. This is where the problem becomes as far as sensitivity goes. So you get twice the selectivity, but you get 1/4 the sensitivity. So it just depends on what that issue is. If your issue is I've got a lot of interference, you would want to use an MSX experiment. If your issue is I need a lot of ions, I need to get good quantitative measurements, and I need a lot of precision, then you'd want to use this wider window, but I don't have a selectivity problem. Okay, so how the heck do we analyze this data? I'll just briefly skim through some of this because it'll be talked about a lot more by other people. So in our lab, we've been kind of trying to refer to this general approach as a peptide-centric analysis. So basically, historically in proteomics, we've taken a spectrum-centric analysis where we basically take spectra, try to search them against a sequence database, use either database searching, de novo sequencing, or library searching. It doesn't really matter what approach you take. It's basically still a spectrum-centric approach. In our case, we want to take peptides and try to find it in our data. And of course, Skyline, FTARM, which is a program that Jimmy Yang wrote for Jim Bruce's lab, does the same sort of thing. Open Swath, PeakView, they are, of course, great example tools of going through in this sort of peptide-centric analysis. In our case, we tend to do it as far as in chromatograms. We have a score that we calculate, etc. We can calculate p-values and q-values just like you can with a spectrum-centric database. And it's ideally suited to data-independent acquisition, although we have been actually applying some peptide-centric analysis workflows to data-dependent acquisition still. So here's this DIA data in practice. Again, here's the data. It's just noisy. There's lots of things. Here's the precursor ion chromatograms, the fragment ion chromatograms. We can point out, here's the point in time, which is our peak, which is not that obvious. And so the current status is we require DDA runs. Either we interspace them with our DIA runs, or you can build libraries with it, like was mentioned yesterday. It can be still labor-intensive peak verifying, and you're limited only to the query space currently of DDA data, which I think I've already shown with the SRM data is less than ideal, because a lot of the best responding peptides by SRM or DIA are not necessarily the ones that are best responding or give you the best signal by DDA. So Brendan has talked about the fact that we've implemented what we believe is the state-of-the-art in this in Skyline. It doesn't use all the capabilities that are in Open Swath, but it uses most of them. And probably the best thing, as he showed yesterday, is that we're performing similarly to those other tools. And there are some data sets where PeakView and Open Swath is slightly better than the Skyline with the mProfit features. And there are some that Skyline does slightly better with than PeakView. So it just depends on the data set. But in general, we're basically competitive with the existing tools. Okay. So what I'm going to talk about just for a few slides is where I believe things are going. This is not available yet. It's going to take us some time to make it available. So, but it is something I'm pretty excited about, and I think it's got a lot of potential. And we're trying to figure out a way to make this usable by a large number of people. So basically, we want to solve this problem. We want to be able to find peptides that were not found by DDA. We want to be able to find the ones that have never been seen by DDA. And so this led into the development of a tool that Sonia Ting is calling Pecan. And basically, the idea is to take peptides of interest, and we have a basic series of scoring, peptide processing, background estimation, peak picking, and some statistical analysis. But the idea is can we come and say that in this data, can we say that this is the retention time that is our target peak of interest? And we've evaluated performance in a lot of different ways. The first way was actually using a manually verified set of data sets that Jared Egerton had done. So there's 204 query peptides from 6 runs, so there's 1,200 peaks. And so of those 1,200 peaks, what fraction can pecan find automatically? And basically it's about 90% without any user intervention. It's 10 master white isolation windows on a Q-Exactive. The second thing we used was actually the Swath gold standard data set, because it's the only published data set out there yet that's kind of used, that's got some manual verification. And, you know, we do okay in water, you know, but, you know, when as the mixture gets more complicated, at the lower abundance signals, we don't have as high a frequency at being able to pick the correct peak assignment. But I still think this is pretty positive. Even down to low atomals, we're able to pick about 80% of the peaks correctly in a relatively simple mixture. Now, this is where I think things get exciting. So if we took a mouse heart proteome and we did a DDA experiment, and we had a percolator, and there's 3,800 peptides in this experiment that we find, and this in mouse heart, unfractionated, and we have a DIA experiment that's less than half the total mass range there. And we said, okay, of those 2,400 proteins, there's about 74,000 potential peptides. How many of those can we actually find? So when you actually look at the data, pecan finds about 70% of the things that were found by a shotgun type discovery type workflow. But we find, and there's an additional 30% that we don't find. But there's an even greater set that pecan finds using DIA that were not found by the shotgun discovery experiment. So while we overlap, we get about 70% of the things that are found by discovery experiments. These ones over here are additional peptides that we find that map back to the same gene products that were up here found previously. It's just a different set of peptides. And this, again, kind of falls into what we've been seeing with using the recombinant proteins, where when you make the protein recombinantly and measure the signal intensity, the ones that gave the best, that we sampled most frequently by DDA runs, were not necessarily the ones that gave the best response. So these are the ones that give slightly better response in a targeted type method. And these are ones that sometimes don't chromatograph very well. They smear at the beginning or the end of the gradient. They have really wide peaks. They're not very ideally suited to a targeted experiment, so that we just don't find them very well in a chromatographic-centric type analysis. Okay, so this is kind of cool, and this is something we're still working on. We're doing a lot to validate these extra 4,400 peptides. But then here's another thing that I find is pretty exciting. So if you said, okay, in this heart proteome, there's 1,200, in theory, mitochondrial proteins, and what fraction of them can we find? We can basically find all of the ones that a DDA experiment found, basically 99%, and there's an additional 100 that we find only by DIA that we would not find by an discovery technique. So one of the things I'm just trying to point out is a different way is that if you do these experiments in kind of a chromatographic-centric, refined approach, where you start with chromatograms, extracting peptides, and use that as your benchmark, and not the DDA results, then we can often find things that we couldn't find otherwise. And you can use these things in order to find new things that you wouldn't find otherwise. Okay, so I've got 2 more sections here. I know I'm kind of probably getting a little bit short in time, but I wanted to highlight something that Brendan's been working on, and I think he's going to try to demo for you today. It's this thing that kind of highlights a little bit one of the dirty little secrets we have in this data independent acquisition field, and that is that with SRM, the data file sizes tend to be small. So if you're talking about using Skyline, it takes pretty much seconds to import these data files. If you're talking about data independent acquisition, you may be only interested in 100 peptides, but you're collecting a ton of data. And if you're not really interested in those 100 peptides, it may be worth it down the road to actually just go back and collect SRM data, just because the analysis per bottleneck isn't much of a challenge, because these data file sizes are huge, and unless you actually really are going to go back and reanalyze the data, it might not actually be worth collecting data independent acquisition data. The import time can take, seconds to tens of minutes per file. So if you're thinking about a case of 50 runs, you're looking at sometimes 10 minutes versus 6 hours to import this data from a very large data set. So this is definitely a challenge, and it's something that most people don't talk too much about in the data independent acquisition field. And so, I mean, you can collect a lot of data in 6 hours. The fact that it takes 6 hours even to import the data into Skyline is a challenge. And this has gotten, it's not a problem with our end. It's getting access to the data files, the chromatograms, fast enough. So about a year and a half ago, working with a close friend and colleague, Nate Yates, we started sitting down and dreaming up this idea, this Chorus project. And so he's at the University of Pittsburgh, we're at the University of Washington. We had initiated collaboration with Amazon Web Services, and another close friend of ours, Andrei Bodarenko, who started a small company at that point in time called InfoClinica. We decided that we'd work together to build this project called the Chorus Project, and I encourage people to check this out. It's an exciting new project. It's only been around for about 15 months now, but it's gaining a decent amount of traction. So in 15 months, we've had very little features or capabilities, but yet we've still gained 764 users. There's been more than 10 terabytes of data been put there. We're now adding about 1.5 terabytes per month. So it's actually escalating, increasing the rate with people putting data there. There's about 200 labs using it. And so it's definitely pretty exciting to see this grow. And even the other aspect of this is that there's greater than 2 terabytes being downloaded per month. So one of the things I'd always been worried about is a lot of these repositories end up becoming sort of data graveyards. But this clearly people are putting data in there and taking it out. 1/5 of the data is being pulled out every month. This is actually pretty exciting from our perspective. And yet we don't even have many tools for people to do that yet. So we wanted a Google-like UI for uploading, sharing, and making mass spectrometry data possible. Automatic account generation, user confirmation. There's extensive security. We spent a lot of time thinking about it. It's basically a laboratory-type model, so we want to make it so that there's the same security that you would have on the same kind of instrument computers in your lab as you would have in the cloud. And data are uploaded as raw files and cached in a distributed data structure. And so we can instantaneously random access thousands of chromatograms simultaneously because it's not a single raw file anymore. We've broken the file up into lots of little chunks. So we can actually have hundreds of processes accessing the data simultaneously now. There's chromatogram and spectrum viewers, and there's a lot of extra tools kind of coming, like database searching, etc. So what do I mean by a Google Docs type interface? Well, this is my Google Docs, a Google Drive interface. We use this a lot to kind of write manuscripts. Use like Jarrett's Nature Methods response right up there to the review. And we kind of use this as a first pass because multiple people can edit documents at the same time. We kind of wanted the same sort of thing in the cloud for mass spectrometry data. You can look at the same thing. You look at my projects shared with me, public projects, etc. And you can look in there. You can orient your files by projects and experiments, etc. You can view the data files without downloading it. It's completely vendor neutral to look at chromatogram and spectrum viewers. There aren't that many analysis tools up there yet, but it's still pretty good. I mean, you can now go, if there's a public data file there, you can even go and look at the chromatograms before you even want to download it to do anything with it. So this is Brendan's idea, and he'll talk to you a little bit more about it later today. And that is that you start with Skyline. Skyline requests extracted ion chromatograms from the cloud, and then Chorus generates chromatograms and returns to Skyline cache. And the question was, could it do this faster over the internet than it could with the data file stored on an SSD drive stored locally on your computer? And the answer, I think, is yes. So here's basically the way it works. Here's Brendan's desktop in the lab, and here's his laptop that just died. But basically, both of these computers are very powerful computers. But here's his benchmarking. So if you have 300 transitions or 1,000 transitions, here's Chorus, this laptop, and he's actually started to even cripple his network connection by tethering through his cell phone as opposed to using the University of Washington internet connection. When you start to get very large number of transitions, it gets to the point where the tether through his cell phone becomes a little slower than that of going through his desktop alone. But he actually did another benchmark where he just did it then through his laptop alone, and of course still, even though his laptop is a very powerful laptop and still got decent memory and a fast hard drive, he can still go way substantially faster to read that data through Chorus on his laptop through his cell phone than he can actually through just his laptop and the data stored locally. So this is pretty key. If you now have gigabytes of data files, you don't want to store it on your laptop. I mean, most of us only have like 500 gigabytes of storage on our laptops anyway. And if you've got these experiments with 100 gigabytes in there, you want to store them someplace in the cloud and be able to access those data files remotely and to be able to process them quickly. So I think this is definitely a feature. And there's one last thing I want to talk about, and it's actually nothing really to do with software or DIA, but I think this is someplace that we need to be investing more effort on is our nanoLC. As we shifted from going from DDA type experiments to targeted experiments, the challenge now for most labs is actually not the mass spectrometry. It's not the data analysis sometimes. What makes the data analysis a challenge is poor nanoLC and poor spray stability. And so nanoLC is a common weakness in most nanoLC platforms. So we've been working for about 3 years now with a company called Core Solutions to try to solve this or try to address some of these problems. And basically what we've done is tried to separate the emitter from the column from the trap, because the trap and the emitter are the first things to fail. And we wanted to be able to use a very smart type interface that if things begin to fail, that it could actually fix the emitter and actually fix the trap on the fly. And so working with Core Solutions, we've built a robot system where we have a cartridge with 4 different emitters. Here's the column cartridge and the trap cartridge. They sit online in a robot. It's basically like a big torque wrench, and it sits and pushes everything, makes all the unions and connections together. Here's the emitter changer. And so now we've also interfaced this with Skyline Runner, so we can make QC measurements on the fly, and if things are failing, we can actually shift the emitter currently to see if that improves things. And I'll have a quick little video here, where basically we're disengaging from the emitter. The emitter changer will be changing to the next position or to a different position. It syncs itself and homes itself, and it can change to any of them. And then it will eventually go back and remake the ultra high pressure seal again on the ferrule. And because there are no twisting and making connections, the connections are actually much more stable than they are even just by a human making connections. The next generation we're currently working on actually also has a trap changer. And it's been a pretty exciting project. It's been a long road. There's a lot involved, a lot of moving pieces here between the hardware and the chromatography aspects of things. It's actually performs extremely well between emitters, between the ports, positions in the emitter. It's using the same emitter but different positions. We basically have extremely good precision between positions. It's not that worse than the worst case that's measured at any one position. Our replicate retention time reproducibility is incredible between position, and you can even go between emitters. We're not doing that much worse than any one of the emitters. And again, the CVs between different emitters are not that bad either. So this has actually been pretty exciting. There's still a lot of work to go there. So but just to kind of give you a sense of what we're beginning to think about. When we originally wrote the grant for this, I kind of pitched this idea to the that you really want to be able to have it so that you got a piece of software could send you an email saying that your run failed in the middle of the night. We're going to try to fix it, and then hopefully by the time you woke up, you got another email saying we fixed it. Don't worry, and here's what the progress is. And so that's basically the idea, is that if it can't fix it, then it would send you another email saying we've halted the run. You need to come in and deal with it, because that's more important than actually continuing to make injection on a failed system. So in conclusions, we've definitely used an empirical refinement to select peptides. We've focused a lot using SRM or targeted data itself to choose our peptides. I think modern hardware and data analysis strategies are beginning to make data-independent acquisition a realistic solution. When we started thinking about these things in 2002, 2003, it really wasn't a good option. We didn't have something like Skyline at that point in time. We didn't have instruments like the 5600 and the Q-Exactive, right? So it really has been a game changer, those combination of things. Method setup is much simpler for DIA than more traditional SRM target methods. No need for scheduling. Although we've done experiments actually where we've scheduled and moved the sliding window of masses up through time. So tends to be lighter, smaller molecular weight things tend to have be more hydrophilic than than the higher molecular weight ones, so you can actually slide your DIA windows across your gradient, which we've done. Being limited to only peptides I've seen previously is not only impractical, but won't use DIA to its full potential. We're working on this. Pecan is a starting point for this. It's a little bit computationally expensive right now, and we're trying to come up with a hosted solution as a stopgap. But that's something we're still working on. We have been working on auto-correcting a nanoLC system, and I do think down the road that all DIA data is going to have, all mass spectrometry data in general is going to be stored in the cloud. But we still, just like with your cell phone, you don't want to go through the browser for everything. You want to use an app that accesses data in the cloud and then displays things in an app. And I think that's a way in which a lot of things can work with Skyline, because these cache file format that Brendan's come up with is actually really efficient to read data from. It's actually really efficient actually to download from the cloud as well. And I think he's kind of shown that from the idea of 20,000 transitions, through his cell phone. He's actually able to download it faster than what he can read the data from his actual raw files. Anyway, so there's a lot of people that did the work here. The current refinement approach, the data that wellness assay is being done by Jim Bollinger. Jared Egerton came up with the multiplexing and the demultiplexing initial work. Most of the mass spectrometry data that's been shown here was collected by Rich Johnson. Of course, Brendan, you all know. Huilin has been working on this the self-correcting nanoLC system, along with Tom Corso at Core Solutions. Nick Schulman is just an incredible developer who's been working on this cloud interface, among many other things in the lab. Sonia Ting has been working on the peptide-centric searching. They actually didn't talk about the stuff that Han Yin's been doing. She's been kind of working on this project that's kind of a mix between DIA and DDA. Basically, it's a DIA that drops windows out dynamically on the fly. It's actually pretty cool. I didn't talk about that today. It's still not quite where we want to be at, but it's growing. Andrei Bodarenko, Christine Wu, and Nathan Yates have been people working at Stratus Biosciences and doing. We've got a very small. This is a project, a budget as far as making Ging Chorus off the ground, but yet we've still, I think, gotten a fairly good application quickly. We've gotten a decent user base already, and it's been pretty exciting to see it grow. Andy Hoofnail and Clark Henderson, when we started thinking about this dried blood spot assay, they are truly clinical chemists. They do, I think Andy once told me, I think there's like 2,000 mass spec runs in his lab for patient samples done per day. And it's pretty exciting and extensive, the type of workflows they have. Most of them are small molecules like vitamin D, but still, these are pretty big, high throughput labs that are being, and they really know what it takes to collect mass spectrometry data and feel confident enough that you can give it back to a patient, to a physician, and not have that physician make an error and cause harm to his patients. So that's something we really have been working very carefully with Andy Hoofnagle and Clark Henderson on, because it's something that we just don't have that much experience with. And a lot of our work is done in collaboration with Thermo Fisher. There's numerous people, but these are just some of the major players. We've got some. We've been really fortunate and lucky to have some federal support from the U.S. government, and also we're really fortunate to have vendors contributing to Skyline development now as well. And there's now 5 major vendors contributing. And we've been told that ABSciX has committed to contributing to Skyline development in the upcoming year. And here's a really outdated photograph of the lab. And thanks for your attention.